Let's do a quick review to the Gram stain. So the Gram stain developed in 1884 by Christian Gram. It differentiated the bacteria into two groups, Gram positive and Gram negative. Gram positive stain purple and Gram negative stain red with Gram stain. So the principle involves it is based on the bacterial cell wall structure. As we know, Gram positive bacteria has thick cell wall about 80 nanometer and Gram negative bacteria have thin cell wall about 2 nanometer. So the thick cell wall contains thick peptidoglycan layer which retains the purple dye, and the thin cell wall loses the purple dye and takes up the counter stain that is red in color called safranin. The purpose and usage of Gram stain involves it is used for identification of bacteria and it guides us for selecting antibiotic. The major disadvantage while using Gram stain is that it requires 100,000 bacteria per ml for visualization under microscope. This amount only gets when there is large amount of bacteria present in the sample, which gets after incubation the bacterial sample in blood culture bottles for 24 hours, and after 24 hours we can get we can do gram staining plus the biochemical test, etc. So the sensitivity of gram stain is low. We cannot directly do gram stain on a sample, with an exception of meningococcemia, a disease caused by Neisseria meningitis, in which there is high concentration of Neisseria meningitis in the blood. There is no need of incubation of the blood culture bottles for 24 hours. We can do direct gram staining on the blood sample, and we can get this range, up to this range and above this range. Now, the procedure of gram staining. We will take the sample on a glass slide that will contain both gram-positive and gram-negative bacteria. The slide will undergo heat fixation, then we will immerse the slide in crystal violet stain for 60 seconds. That will stain purple both the gram-positive and gram-negative bacteria. Then we will again immerse the slide in Lugol's iodine for again 60 seconds. Lugol's iodine acts as a mordant because it strengthens the interaction between the crystal violet stain and the cell wall. As we know, the cell wall contains peptidoglycan, and by adding the decolorizer, the gram-positive cell wall will not lose the crystal violet stain because of thick peptidoglycan layer, while the gram-negative will lose the crystal violet stain because of thin peptidoglycan layer. So by adding the counter stain, that is safranin, the gram-negative will appear red. It will take the counter stain while the gram-positive will remain purple. As you can see, the gram-positive appear purple while the gram-negative will appear red with the help of gram staining.